today, and for all of you for joining to learn a little bit about pest reporting throughout Hawaii, and specifically about the project that I work on, 643pest.org, and have worked with for 15 years. I am a staff member with the Hawaii Invasive Species Council, though I work for the Pacific Cooperative Studies Unit at UH Manoa. That's one hat. I also have another hat. I'm based on Maui, and I also work as a community liaison with the Maui Invasive Species Committee. Oh, there we go. And there goes the moving. If any of you guys are old enough to remember this. So I think that all of us that are here today are interested in knowing about what diversity of organisms are found throughout the state. Not only do we want to know what is out there, we want to know where they are across the state, what the populations of these organisms are, and especially of interest to many of us because there are 30 potentially invasive new organisms that come every single day to Hawaii. We're interested in knowing about any of the new high-risk invasive diseases, invertebrates, plants, animals that are showing up somewhere in the state. And a lot of us would like to be able to have the public and their eyes and ears helping us to find these new high-risk invasive organisms. And optimally, they would be finding those and letting someone know about it during that window of opportunity for eradication, which is quite early in that whole invasion. So luckily here in Hawaii, the residents of Hawaii are actually pretty aware of the concept of invasive species, and they consider invasive species to be a problem throughout the state. The Coordinating Group on Alien Pest Species, CGAPS, they have organized a number of public surveys of statewide residents all the way from the late 90s through to just a couple years ago. And one of the questions that they would ask is about the likelihood that this resident, would they report a snake if they saw it? And if you look, that's incredibly high. Almost every single person that they asked said they would be likely to report a snake if they saw one. And if you talk about something that maybe isn't as scary as a snake, like an unknown insect, you're still looking at about 50% of those respondents to the survey said that they would be likely to report an unknown insect. So that's working on our side if we're asking the public to help us look for all of these new organisms. Well, then they also went further to ask, so if you saw a snake, who would you call? Now, if you notice the survey response, that is the purple one on the left, that was conducted in 2004, and fully a third of the respondents said that the first place they would go to would be the Humane Society. And you can look at the variety of other answers that were given in 2004. In between that, the survey responses on the right and that first one, there was targeted outreach throughout the state to tell people what to do if they saw a snake or how to report pest. And if you see, there was a change. Police and 911, which is the correct place to tell people to report a snake, that really rose up to about a third of the respondents. But if you go down that list, there is a huge variety of different organizations and groups and a lot of people just didn't know, and they thought that they would just have to look it up. So further complicating that, and I think that everybody here is probably aware of that, throughout the state, there's a whole bunch of different agencies, groups, organizations working on invasive species, each with their own internal methods for dealing with pest reports from the public and from partner agency. And these systems and communication networks, whether they be formal or ad hoc communication networks, often vary from island to island, and they often vary even between same agencies from island to island. So what can we do to make it easier for the public to be able to do what they really want to do and communicate about these invasive or unusual organisms that they're seeing throughout the state? How do we ensure that the right people are getting these reports? And when I'm saying the right people, optimally, we want the people that are going to have the capacity for rapid response to the organism being reported. We want those reports to go to the people that can identify these organisms, as well as be able to conduct, if needed, a risk assessment so that when they are seeing this pest report, they can ID it, They know whether or not that needs to go into the hands of another organization. And then finally, how do we share this information amongst the invasive species community? And shout out to Roshan and these invasive species mini conferences, because I think we're all here today for that exact reason, to be able to share this information amongst our community. So one such approach to answering those questions is the 643 PEST phone hotline and 643pest.org online pest statewide hotline. So just to clarify, the pest hotline phone number is actually a Department of Agriculture project. It started in 1992 with 586 Pest, which was 2 lines in Oahu that were taking phone calls from anybody in the state about pests that they wanted to report. And they said the second half of that down, those pest reports were getting to people who could identify them and who had the capacity for rapid response when necessary. But there were some issues. If you called from any other island besides Oahu, you incurred a long distance phone charge. And as well as that, in those surveys that CCAPS was conducting throughout the state, they found that people strongly wanted to talk to someone on their own island about what they were seeing. Somebody who was familiar with the locale and with the situations. So in 2006, that 586 PEST hotline was updated to 643 PEST, which is a kind of smart phone number. It's the same thing that Pizza Hut uses. It allows for when you call that number, your phone call will be routed to a Department of Agriculture office on your island for most of the hours of the day. Those calls are answered by a person, often a plant quarantine inspector, except for just a few hours at night. And in 2007, there were statewide promotions on TV and radio, and a snazzy little Frank DeLima song was made about it to promote it. Around the same time that the pest hotline with the phone hotline was getting started, there was a separate track with different agencies wanting to start an online pest reporting system that would be really similar, cover the whole state, be able to get those reports to who they needed to. Started with a proposal, the pilot multi-agency early detection reporting system that started in Maui County, and that original proposal was written by Dr. Lloyd Loop, a USGS scientist based on Maui, as well as another USGS group, the Pacific Basin Information Node, which I was employed with, and HISC funded it in 2005. We created a website called reportapest.org, a backend SQL database, and we had a vision to, oops, let me go back one, sorry, to stimulate early detection, so to actually get folks reporting organisms when they were still in that window of early detection and rapid response when we could get them, to make it user-friendly and have rapid assessment of reported biota and ultimately facilitate that rapid response. At the same time as it began to develop, we realized that was really important and became part of our vision to be able to have a distributed workforce that could respond to these pest reports, including people from many different agencies and groups across the state, so that this wasn't something that was just limited to one agency with one geographic location that it tended to mostly focus on. In 2015, we rebranded Report a Pest to 643pest.org so that we could co-promote with the Department of Ag's Pest Hotline. And you can see on the right, that is it's a pretty basic, there is a pest reporting form. We did a couple years ago also made Android and iPhone apps. And I have not verified this, but I choose to believe it really is. But Governor Ige said that this was by far the best pest invasive species reporting app in the state. Behind the scenes of all of that, we have a model where we have one person, a facilitator, which is my role in this, one of my roles in this project, in which we want to help people along throughout this whole process so that people are feeling heard. We're able to get all the information we need to be able to identify and know the location of these organisms being reported. We have a whole number of different specialists, both taxonomic and location specialists throughout the state that can help us with identification. We work with a whole bunch of different responding agencies and groups throughout the state and update our contact lists every year so that we know that we're getting all of this information of interest to the right people for that rapid response. And then to close the loop optimally, we're letting that original reporter know about the organism and what happened to it so that they're feeling that they're being heard and they know what happened. So all of the facilitators, we're not all entomologists or botanists, but we all have a training that we go through so that we know 40 to 50 of the most commonly reported species, as well as look-alike species for target organisms throughout the state. So that we are able to effectively filter out a lot of those common species reported and save time for all of the people that are actually out there doing the rapid response. So things started off really slow, and it's actually a little bit more than what it shows on here, but it has been building over the years to the point at which we have 430 pest reports for just this year. And from that upward movement of these or the increase of reports over the years, we're looking to increase by 100 reports every single year that we can process. Most of these reports, you see what this diagram is, we really make one of our primary focuses, along with communicating with the reporter, is we really try to make sure that we get whatever organisms are reported identified. And for any of those of you that have looked at blurry images of insects or read kind of crazy descriptions of things, you know that can be sometimes hard, but we work really hard at that. And the majority of what is reported, we do identify. Over the years, we've had 275 unique species reported and identified. And where are these reports coming from? So believe it or not, most of the reports are coming from Oahu, and that's probably because that's where most of the population is. I think that of note here also, during those CGAPS public surveys of residents, I found it pretty interesting and consistently throughout the years, that neighbor island residents were often more aware of invasive species, of specific invasive species issues like rapidohea death, specific organisms, and they tended to think of invasive species as a bigger problem in the state than did Oahu residents. And I think that you find that reflected in the quarter of the reports that we get also from Maui and the Hawaii Island, from Big Island. Kauai, not so much, and I really can't speak to why that is entirely. Another piece of background, we haven't done a whole lot of big promotions for I guess either the pest hotline or online pest reporting in a while. And from what we see from our web analytics, we get nearly 40% of all of our traffic comes from links from other people's web pages, from partners. And then maybe another 20% or so are people that actually enter the name 643pest.org into into their browser. So there's always room for improvement, and that also could be focused in specific target areas if we wanted to improve the number of reports we were getting from those areas. So you can see the pest reports from across the state, and it pretty much correlates with where people live, where they work, where they play. And you notice that almost all of these, I think 99.99% of them are terrestrial. We can accommodate ocean invasive species, but currently in that space, there are other tools being used. But we primarily have a terrestrial and freshwater aquatic focus. And just a close-up on Oahu so that you can see where those pest reports are. And as you can see, the vast majority of them are along roads and people's backyards, parks, and then a few in hiking areas. And before anybody freaks out about it, a lot of those super remote ones are hikers, and they often are for things like Jackson's chameleons. So the types of things that people are reporting, well, it actually tracks a lot with what we saw earlier with what people were interested in reporting, those snakes or vertebrates and insects. So roughly half and half of our reports come from vertebrates and invertebrates. Of those vertebrates, a lot of them are herpetofauna. We get a lot of reptile and amphibian reports. And with the insects, we get a lot of stinging ant, other pest insect reports. But to me, I always found it interesting as a plant. I like plants, and there are a lot of people throughout the state doing a really good job of getting the word out about invasive plants that we're looking for. We get very few plant reports. I'm always excited when we get an invasive plant report. And but it may not be a surprise that there are very few fungi and pathogen reports that we get. Now, of these reports, just about 25% of them are what we call actionable. Those are reports that that are forwarded on to a response agency, either for rapid response and control, for further investigation, on-site trapping, identification, et cetera, or sometimes also for tracking when people are trying to figure out where an organism is throughout the state. That 25% being actionable reports, is anecdotally what I hear is the exact same situation for people across the state, like from the Department of Agriculture, the invasive species committees. I'm hearing a similar kind of spread of just about 1/4 or less are actually organisms that are of interest to us to take action on, and that a lot of what is reported are what we consider to be widespread invasive species. So to look a little bit closer to see what we're seeing by island, I went on and put together what the top 5 species are for the main islands that we've been getting pest reports from. Starting with Hawaii Island, as you look on here, a lot of these organisms, little fire ant, koki, myconia, on the Big Island, there are no island-wide control of those organisms. They're considered somewhat widespread, though they are not throughout the whole islands. And you'll see these guys, Jackson chameleon and mongoose, they'll pop up again. I think people go online to try to figure out what they were seeing and then see it says invasive species and feel like they need to report it. On Maui, it's a little bit of a different situation, but koki is absolutely #1. For those of you that are not aware, there is a koki population that is somewhat sizable on the slopes of Haleakala in the Haiku area. Those koki, though, are still a pest that is responded to. So they are not widespread. So but there's enough of them that people hear them, and people are pretty aware. There we go. Thank you. Now I'm unmuted. We also get a lot of tropical fire ant reports, not only because they're bothersome, but I think also because there is a lot of word getting out about little fire ants. So when people encounter stinging ants, which is a really good thing, they know to tell somebody about what they are seeing out there. On to Molokai. There has not been many reports from Molokai, but it is a little bit interesting that, and I think that's because it's a factor of the size, the factor of the fact that the land managers, the people doing invasive species work are really good at communicating with people and people know who to talk to. But we did get one, they actually, Ficus religiosa is, the bow tree, is a target for the Molokai Invasive Species Committee. They are eradicating that tree throughout the state. On Oahu, we see a whole lot of oriental flower beetle reports, greenhouse frog reports, tropical fire ant reports. All of those are lookalikes for target species on Oahu. There is a lot of word out about the coconut rhinoceros beetle, so people are out looking at, and also the oriental flower beetle is it's spread pretty fast across the state, and it's a very noticeable insect. And then finally on Kauai, as you notice, way fewer, and this is where one of the big caveats of what we do is coming through. So you see that there's 11 mongoose reports. Not every one of those was a mongoose. And we do have, when we pass off these pest reports to the response agency or group, we do not always hear back about what they found out. So that is a caveat, and we do record data within our own system in order to be able to weight those species identifications, so that it is, as you're doing data analysis, there are ways to be able to weight that out, that they aren't all that organism and where they were found. But you can see the rest of them, and as you see, there's way fewer on Kauai. And I think that that is all of the time that I had today. So I just wanted to thank you all. I didn't put all my thank yous, but thank you to everybody that's out there that has worked with us over the years. And I would love to hear more from people if you would like to know more about this project, or if you would like to, sorry, I just noticed a typo there. If you would like to work with us, or if you feel like this is something that you should be involved with.
Thank you, Elizabeth. And we have time for questions. The participants can unmute and ask question. I don't see any questions for Elizabeth in the chat box. So yes.
Hey, Elizabeth. I was just wondering how often you get reports of dead and dying trees. We get a lot of those calls, and I bet JB gets a lot of those calls too. And I'm wondering if you get them, and if you do, what your protocol for that is.
So thank you for the question, Rob. We don't get a lot of dead and dying trees. It kind of falls in with that same, we don't get a lot of plant reports. We do get some, and I think there could be a number of reasons for that. Like I said, we get the majority of our traffic from cross-linking and cross-promotion. So it just may be that a lot of, especially with rapid ohia death, that there are other mechanisms for reporting that are being promoted. But the procedure that we do thus far is I try to follow along with so I guess dead and dying trees is multiple things. With ohia trees, there is that process that's been set forth for the different groups that are actually going out and following up for ohia trees. For other, I do get a lot of palm trees and palm damage trees. Usually people think that those are coconut rhinoceros beetle damage. And actually in that instance, I will share it with the coconut rhinoceros beetle team on Oahu, but I do not share it beyond that. So, and I do, and I guess it's of note, as this is a CTAR presentation, I do a lot of times with things that seem a little bit more esoteric, some kind of palm disease, some other type. I send people most often to CTAR for diagnosis of their backyard plants issues. Does that kind of answer your question, Rob? And if you want to leave a comment for a better place for me to be sending those, please do.
Oh, that's great. Thanks, Elizabeth.
We have still time for more questions. And I see comments from JB saying he got calls about dying trees. And usually these trees that our landowner had girdled or herbicide, and someone driving by sees them, and he seldom follow up on this.
And I don't have a question so much as a comment. I just want to say that we, working on the two-line spittle bug, we were looking for a way to do reporting. And so we've worked with 643 Pest, specifically, getting this all updated on the beetle, the bug and like where it's at. And, so that she had a bunch of information ahead of time. And then we've been doing in our promotions to get people to report that insect, which we kind of started towards the end of the season. So we don't expect that many reports actually this year, but it was an opportunity. If you have like a new thing that you're looking out for, I think reach out to Elizabeth and, let her know you have this thing, and then promote 643 Pest as an easy way for people to have one place to report. Like take advantage of it, I guess is what I want to say, because it made things a lot easier for us, that people aren't getting an answering machine or a voicemail while someone's in the field, and maybe you're going to call them back, or it's kind of confusing, whereas this is just one central thing that people can use. And I think it's a good service for new pests.
Well, thanks, Franny. And I definitely am, we are willing to, and we have the capacity to handle more pest reports. It is not just me answering these reports. There are people from other agencies and across the state answering the reports. But we are we love working with other folks to act as a conduit for those reports. And we can save you a little time because we can figure out if it is obviously one of those meadow spittlebugs or some other common species. We can save you the time for communicating about that and get you the reports that are most likely to be your target.
Yeah, that was one of the big things for us was the screening, right? So it takes out, I know, the researchers on this project are so overwhelmed, and having that screening done, like, that's really awesome. Really appreciate it.
Okay, yes.
Yes, okay. My name is Jia Wei. I'm from Department of Pests, and I'm originally from Malaysia, which is also a tropical region like Hawaii, which share some of the similar tropical pests. My research looked at the ecology, physiology, and behavior of urban insect pests, and essentially the development of improved pest management strategies against them. In this talk, the termites that I will cover is mostly invasive drywood termites, not subterranean termites, sorry. But I will share with you the differences between these two termite groups. Then I will proceed to invasive pest ants and talk about a new management strategy. So I'm gonna start with good things about termites. Termites help in degradation and decompose by breaking down cellulose. They are called earthworm of the tropics that aerate the soil and mix nutrients, returning carbon and nutrients to the soil. They are also food for birds and reptiles. There are about 3,000 described species of termites. And I want to show you that not all termites are pests. There are so many termite species in the world. Some are just hanging around in the forest, but those small percentage here, they are nasty termites. Some mound building termites are very good architects. This photo is from my former PhD advisor. Mound building termites are not found in Hawaii. There are good and bad termites. The bad ones are very destructive insect pests. Not only they eat wood and eat our homes, they also eat cash. The termites don't care what currency it is. They are hungry insects and they eat everything made of cellulose. They also attack cork. They are actually not interested in wine, only the cork. Termites occur in all 50 states except Alaska and most common in Hawaii and the southern states. They prefer warm and moist environment. Hawaii is one of the states that has the most serious termite infestation in the US, unfortunately. You can see that Honolulu is in the top of the list, top 50 termite infested city. In addition to Oahu, of course, they can be found on almost every major island in Hawaii. While most termites only attack dead wood, there are some termites that are known to attack living trees, such as fruit trees and palm trees. But usually the damage is on the inside where you cannot see what is going on. For example, on Hawaii farm, the invasive termites, subterranean termites attack fruits. Here is a photo of a banana attacked by termites. In addition, they also attack many palm trees. Those photos are from Dr. Scott Nelson. And on Kauai Island, you can see that the drywood termites, they damage many live citrus trees. And thank you, Roshan, for the photos. So in addition to that, termites are known as structural pests. Termites has been reported on Oahu for more than 100 years. From Oahu, it gradually spread to all of the major islands through shipments of infested wood. As termites are very active in Hawaii, it is estimated to cost residents of Hawaii over 100 million each year. Here's a little bit background on termites. They are social insects living in colonies. There are three main groups of termites. The most important termite groups are subterranean termite and drywood termites. So those are drywood termites. Their body are more cylindrical compared to the subterranean over here. And for drywood termite, the head of the soldiers are constricted and highly wrinkled. In Hawaii, the most common and destructive termite species are Formosan subterranean termites, as well as the West Indian drywood termites, Cryptotermis brevis. These two are the most economically important and invasive. The subterranean termites or ground termites is one of the termite groups. They build the nests in soil and cryptic spaces. They are highly depend on moisture for survival. They are also susceptible to desiccation when exposed. When they are tunneling or searching for food above ground, they will form mud tubes to prevent desiccation and protect from natural enemies such as ants. The presence of mud tube is usually the sign of this termite infestation. Those are mud tubes that you will notice around building. This is like the highway or freeway, which is called mud tubes, and the function is to prevent them from desiccation and protect them from natural enemies. Termites like to attack door frame and window frame. Those locations are also called termite hotspot. Another termite group is drywood termites. They live entirely in drywood. They don't need access to soil and less dependent on moisture, and this species is common in high-rise building. The infestation normally occur in wood furniture. They produce holes on the surface of the wood, expel tiny fecal pellet, or also known as frass, that look like tiny grains of sand. The evidence of drywood termite infestation is the presence of these very tiny fecal pellets, known as frass. Frass are usually ejected from kick-up holes produced by drywood termites on the surface of the wood. And you can see here, this is a kick-up hole by drywood termites. In this talk, this drywood termite species is what I focus on. The common name is West Indian drywood termites, and the scientific name is Cryptotermis brevis. It is important termite species in Hawaii. They are invasive and native to Pacific coast of Chile, South America. During COVID-19 lockdown, I've been working together with a local heat treatment company and pest control company to investigate the efficacy of heat treatment and why some heat treatment failed in the past. We went to a very nice condominium at the 40th floor, which was infested by drywood termites. We set up a heat treatment project to treat termites in this kitchen here, which is the infested area. We used electric heaters to raise the air temperature to between 120 and 140 degrees Fahrenheit. The treatment have to start by removing all personal items, leaving the cabinet doors open, put heaters and fan inside, and then covering up the treatment area. This is the infested kitchen before covering up, and this is the look of the treatment zone covered with the tarpaulins to contain the heated air. When doing the inspection at the condominium unit, the drywood termites and frass and termite damage was mostly found at the base of the cabinet. So before the treatment, we have to do some preparation work, such as leaving all the cabinet doors open, cover the air vents, and insulate the fire sprinklers. We experienced firsthand the entire process of heat treatment. This technology is actually also commonly used to control bed bugs. When considering different treatment options for termites, heat treatment is a non-chemical option, so it is non-toxic. The downside of heat treatment is it is quite expensive. So what are the challenges that we faced or have to think about when doing heat treatment? We found that heating the toe cake is a challenge. So what is a toe cake or toe space? Toe cake are this recessed space at the base of the kitchen cabinet. It is a very common design in the kitchen. We investigated why heat treatment failed sometimes and how to improve the efficacy of the heat treatment. One thing we noticed is during the heat treatment process, the heated air cannot go into these toe spaces. This is called heat sink zone, where higher temperature may be needed to heat it. However, the heat treatment is done, if the heat treatment is done for too long, to achieve the target temperature that we want at toe cakes, it can potentially damage heat-sensitive items on the top. But if the target temperature cannot be achieved at toe cakes, the termites over here will survive. So one solution is to drill holes. This is the floor, the blue color tip, and this is the toe space. We drill hole at the back of the cabinet over here with a base with toe kick voids. And we put this tube to direct heated air from a heater into those toe spaces. We put sensors and a probe here in several hard to reach and hard to heat areas to ensure that the temperature was high and sufficient to kill drywood termites. We wanted to make sure that more heat reaches the toe kick voids and subsequently achieve the target temperature. So I know that homeowners will ask, can we not drilling any holes? We can ask homeowners back, do you want to have holes on the cabinet or rebuild the entire infested cabinet? But what should we do with the hole in the cabinet after the treatment is completed and done? My collaborator from the company used a customized round plastic cover to cover up the holes after the treatment. So problem solved. Now back to the inside of the treatment zone. We use a few heaters and fan to generate and circulate heated air. All equipment have to be registered to be used, so they have registration number on them. During the heat treatment, we use temperature sensor here to monitor the ambient and internal temperature of a few spaces. And then real-time temperature data over here was sent to a software on a computer. And depending on factors such as degree of the infestation, structure, material, treatment area, and ambient temperature, the treatment time different among homes and takes from 2 hours up to a few days. In addition to toxicity, heat treatment provides some flexibility to homeowners because they can choose whether the treatment is to be applied to the entire home or only to known areas of infestation. This is different than the fumigation or tenting. Another benefit of heat treatment is the occupant can move in immediately once the heat treatment is completed, as compared to fumigation where the treated structure needs to be aerated and vacated for at least 48 hours after the treatment. So this is another method, fumigation or tenting. This method is recommended for severe or widespread infestation. Fumigation is not possible to be done only in a single unit in high-rise building, just like the condominium unit that I show you and I talk about. For fumigation of tenting, the entire building, which means every floor of the condominium, has to be involved. This is the last slide before I move on to ant control. The follow-up inspection is ongoing to confirm activity or inactivity of direct termites after treatment. And we use a termite detector system to monitor that. We are in the process of publishing this information to inform the pest control operators and homeowners the termite control option they have and the things they have to consider to improve the efficacy of the heat treatment. Now let's move to another topic, which is ant management using hydrogels. When I was a postdoc at UC Riverside, I developed alginate hydrogel beds along with 3 other members in 2017. Hydrogels is a super absorbent polymer that can absorb a lot of water. So it can be used to deliver liquid bait to ants without using bait station. I joined UH last year and I was recently awarded HISC funding as a PI to continue my biodegradable hydrogel work. The work will be focusing on yellow crazy ants and other sugar-feeding ants. It is ongoing research. We found that hydrogel baits are effective on a variety of sugar-feeding ants. Here is a little video clip on yellow crazy ant feeding. So you might wondering, oh my God, will these hydrogels attract non-target insects such as pollinators? Here is the answer. So the research, a new research done by Purdue University, investigated if hydrogels are attractive to pollinators at ground and platform placement. The result shows that common pollinating insects such as honeybees, solitary bees, and butterflies hardly visited the beds and accounted for only 3%, about 3% of all visits. He concluded that the risk to pollinators is relatively low since the majority of arthropods attracted to the beds were taxa that are extremely abundant, such as flies, beetles, wasps, non-sensitive, and in some cases, pestiferous or invasive. So why do we need to control ants? They are not only urban pests, but in agricultural area, these ants form mutualism with hermit-parent pests. They milk them for their sweet secretion, protect them, and often transfer them to young and healthy plants when the old plants die. This is the reason we see outbreaks of skill insects and many pests, mealybugs, with the presence of these ants. Ants are nuisance in urban environment, we all know that. And they are more than just protecting hermit-parent pests in agricultural settings. Ants can directly damage fruits such as bananas by their abdominal secretion that contain formic acid. Formic acid injuries usually appear to be sunken and dark here. And in Hawaii, the ant species that produce formic acid includes those ant species that you can see from the screen. Anoplolepis gracilipes, Paratrachina longicornis, and all of these species here. And traditionally, people have used spray insecticides to control ant population, but it creates a lot of environmental issues. For example, Hawaii is surrounded by water, so chemical spraying causes a lot of runoff issue. Bait is an alternative to spray insecticides, but the bait stations are expensive, the bait spill easily, and they need a lot of maintenance, such as checking, cleaning, and refilling. The hydrogels can hold sugar water until the ants show up and drink it. It is a cheaper alternative to bait stations and make the bait stations unnecessary. We have been producing hydrogel in the lab, and I'm going to show you a video on how I make hydrogels using a shower head. All right. I want to clarify that the bait applying under the tree is just a demonstration, and in real situation, we have to apply the bait close to the ant foraging trail. I used food dye to dye the hydrogels. After the ants feeding on it, their abdominal color changed and the hydrogel becomes smaller. The hydrogels can also get smaller through evaporation. However, it can absorb water or rainwater and become hydrated again. After applying them in the field, eventually they will disappear on the ground because it is biodegradable. We did an experiment in commercial citrus grove that had very heavy Argentine ant populations and severe mid-parent pest problems. So this is a 1 minute visual count on number of ants. The blue shading indicates baseline measurements taken before treatment. At the end of the experiment, after carrying out 3 treatments, we achieved about 85% reduction of ants. This is a close-up photo of Argentine ant feeding on hydrogels. Many people said that they look like boba drink that we are familiar with, but boba cannot absorb liquid and become hydrated or larger. In my research, the hydrogel were produced first and then conditioned and then hydrated in liquid bait laced with a small amount of toxicant. In addition to controlling ants, the hydrogels can also be laced with chicken juice from a can. For flying insects, the cages with hydrogel baits were hung on trees in urban parks to manage yellow jackets without having to locate and treat the yellow jacket nets. And they are super attracted to this chicken juice hydrogel. Other than the boba hydrogels, we are developing hydrogel in a different shape. They look like regular gel bait, but it is hydrogel which is hydrophilic, so it can absorb rainwater and stay moist and remain palatable to insect pests, compared with the regular gel that could dry up quite easily. We are developing this sticky and biodegradable hydrogel that can stick on plants to target other insect pests as well, in addition to ants. And lastly, thanks to those contributed to our research and urban entomology program. It is not possible to run my lab and research without your support. We are looking forward to collaborating with more people in Hawaii or other states. Could be researchers from universities, agency, as well as the industry, and those interested in insect pest management. I'm hoping that the research outcome can be directed or indirectly translated into operational activities for pest managers or farmers.
Thanks, Jiawei. Now we can have, oh, you still have?
Oh yeah, the last one.
Okay.
The last one is on a different note. I'm developing a course called Medical and Urban Entomology for next fall. The course has 2 credits, online lectures, and 1 credit hybrid lab. Feel free to share it with UH students who may be interested. And feel free to send me questions through email or the chat box, or maybe we have a little bit time now. I'm not sure. Thank you for your attention.
Yes, thanks, Jiawei. We can have at least one question for Jiawei, and many more questions in the chat box.
Oh, okay.
So we have time for one or 2 questions.
Okay.
I have a question.
Yeah, sure.
So, did you actually use active ingredients in pesticide when you were? You did, but what was that active ingredient? What was the pesticide that you used?
We actually use a variety of active ingredients. In the past, we tried to use thiamethoxam, but it is a neonicotinoid that may harm the pollinators. So we switched to using boric acid now.
And then a follow-up, is there, how can other people use this, or is it only for research now? And if it is, then is there a possibility of the future of people, other people throughout Hawaii being able to use this technology to control ants?
Right now, it is still in the research stage, early stage, but there are some companies that show interest who may be able to commercialize it. But at this point, we are not really sure. So hopefully in the future, it can be, everybody in Hawaii can be used as a commercial product.
Great, thank you. Great job.
Thank you.
Very interesting.
Yes, thank you.
Okay, so the Department of Ag did a rapid response to the CBB invasion on Kauai. The distribution of coffee bearer borer in Hawaii. CBB was first confirmed to be established by Hawaii Department of Ag and Pest Control on September 2nd, 2010 on the Big Island. It was then found on Oahu in 2014, on Maui in 2016, then in August of 2020. The CBB was detected on Lanai in August. On September 3rd, the CBB was turned in to HDOA by KISS, who received a call from the 643 pest line and from a resident farming coffee in Kalaheo. The beetle was sent to Oahu and confirmed to be coffee bearer borer by our entomologist, Janice Matsunaga. Okay, H2A and CTAR staff conducted a site visit the day after the CVP sample was received. The next day, we started delivering surveys and with CTAR in the Kalaheo and the Y properties and the surrounding properties to determine the extent of the infestation. With the permission of property owners, we stripped trees of the infested coffee cherries that we then bagged them and froze them. Yeah, in our delivering surveys, there were 7 properties to be found to be infested with CVB in the Kalaheo Y areas within a radius of a half mile. And the farthest property from the Kalaheo Y was found in the Y highlands above Kukuila, about 1.3 miles away. And the response consisted of a collaborative effort between HDOA, CTAR, KISC, and Koi Coffee, and the property owners. Okay, in order to clean up the area and use some sanitation methods, we stripped the branches and we removed coffee berries to prevent the CVV movement and to destroy the host material. We also set up hookah traps to attract the beetles that are in the area. See, we cut the trees, we cut the branches off, we stripped the branches and the berries off the branches into rubbish barrels, and we collected all the cherries and froze them. Okay, in a summary, how could we be more proactive in monitoring CVV throughout Kauai? There will be a collaboration between agencies is important due to lack of manpower and resources within individual agencies. And trapping for CVV traps should be set up when coffee is green on the trees. The lures could also attract beetles from other locations. Therefore, the traps could be set up away from coffee fields, about 200 to 400 meters away, and put up along roads with coffee trees away from the main coffee fields. We can see that the spread of CVB did not happen overnight. Our surveys indicate that CVB was established at different locations in Kalaheo area within a radius of 2 miles. And integrated pest management must be used to control this infestation. Okay, Roshan, thank you.
Thank you, Craig. We have some time for questions,
Any questions?
discussions.
Yes.
So we have lots of time for questions and discussion.
Okay.
Yes. So yeah, Craig, how many sites we cleared out among those 9, and how many we plan to do? I think we did 3 sites. Yeah,
3
we
sites,
clear
yeah.
out 3 sites, and we are
Yes.
looking forward to do at least another 3, which is
Another 3 more.
we're just
Yes.
waiting for the permission from the homeowners, right? And
Yes. Yeah.
yeah,
We haven't responded yet.
CVB was detected from 7 sites or 9 sites?
Right now we have 8 sites positive, and we have one other site that we know is positive, but has to come from Roshan, yeah, to be the 9th site.
So currently, right? Recently, 19 or 20? Recently?
Okay.
Did you found in 2020 or before?
9. Yeah, actually we had nine sites. Yeah. We have 2 in the Y within about 100 feet of each other. Yeah, but it's 2 different property owners.
so Sarah, the coffee berry borer was confirmed on Kauai recently. I think it's the first week of September. But
Okay,
we think the coffee berry borer was here from long time ago. That's our assumption, right?
I see.
Yes.
Otherwise, we will not have this multiplying population, right? So
Yeah. Yeah.
until now, we have 9 positive sites around Kalaheo and Lawai area. And we have been successful in clearing out 3 of those owners' property. And we are looking forward to clear out, clean, stripping, whatever you call, in another 3, at least another 3 property. And we are looking forward to that. Thank you. Any questions? So Craig, Randy has questions. Randy can. So Yeah, there is more questions. Monte Tudor Long is asking if there is any prospects of local eradication of CBB.
Eradication right now, no. There's just too much
and
widespread. Yeah.
uh, and Randy has also same questions. Same question.
Yeah, in order to eradicate, I don't think it's possible. It's just too widespread in that, like 2 mile area, and there's just too much coffee trees nearby in the surrounding area. Like above the Clay Hill area, there's a whole forest of coffee trees right now. They're not in full bloom right now, or the berries are not ripe. But there's like thousands of trees in above Clay Hill. Uh, Lower Lawai, there's only Kauai coffee fields, and a lot of people they willing to get rid of the trees in order to help Kauai coffee in the Lawai area. So Kauai coffee is not harvesting their fields below the our find in the Lawai area. They discarded the field there. I talked to John Ching, and they discarded field. For what reason? Could be vine. I think it was vines. Um, but they're still finding fields with CVP. So eradication is kind of hard. Also, like when the harvesting techniques of Kauai coffee, they leave back a lot of beans there on the ground, and they'll have to do a thorough job of vacuuming or getting rid of all the berries that are left behind after harvesting, and it's going to be a real challenge for them. Uh, if they need help, we'll try to help them as much as we can. But anyway, I don't think eradication is possible at this time.
Thanks, Craig. I'm just seeing a chat box. Andrea Kawabata, our extension agent at Kona Big Island, also is in our rapid response team. Andrea, if you like to speak something about the possibility of whether eradication or not, or Andrea?
I will be very blunt and say that eradication is not going to be possible at all.
It's hard.
Okay. Can you tell us why? Just
we don't have enough money, manpower.
Okay.
Thanks, Bashan. Okay. Ready?
Ready, yes.
Okay, Mr. Odontomachus probably would GDS, aka trapjaw ant. Sorry, how you go back? How you go back, Craig?
Back arrow should work. Back arrow.
Back arrow. Sorry, my first time. Thank you. Okay, ants in this genus differ from other ponerim ants found in the United States. Their unique head shape. They have large tapering petiole, large size of the workers, pedicure mandibles inungulate and inserted near the center of the clypeus. Sorry, there's the meanings on the bottom in orange if you folks don't know what those words are. I don't know myself either. They're called trapjaw for mandibles that can be opened to 180 degrees and snap rapidly together on their prey. They have the ability to control and time the mandible movement and forcibly close the mandibles against a surface or other organisms and propel itself away for up to several inches. They can use the mandibles for much more sensitive movements such as caring for larvae or nest building. And I saw them moving with my eyes moving larvae and building their nests. Okay, trapjaw ant behaviors and dangers and threats. The obvious danger regarding trapjaw ants is their bite. Scientists have found the ant jaws accelerate at 100 times the force of gravity. When snapping their jaws shut, trapjaw ants reaches speed up to 145 miles per hour. Oh, mistake that one. Exerting forces 300 to 500 times the ant's body weight. However, these ants also have a painful stinger, and if a nest is threatened, workers immediately jump at the attacker, stinging and biting. Trapjaw ant stingers are not barbed and can be used repeatedly, although their venom isn't fatal, but it can produce an allergic reaction. Building small colonies with only one queen and less than 200 ants. Trapjaw ants eat small insects and sweet substances, but we see them eating peanut butter, so yeah. Trapjaw ants nest in soil, leaf litter, and rotting logs. They are typically found under dead wood or beneath forest floors. In urban areas, their nests can usually be found in mulch underneath hedges of landscape and near homes, commercial buildings, malls, and hotels. Okay, on Kauai, the trapjaw ant was first discovered about June 10th, 2017 by a terra farmer here in Hanalei. The farmer turned in the sample to U.S. Fish and Wildlife, who forwarded the ant specimen to other agencies. The samples were then given to HDO entomologists in Honolulu, who notified us here on Kauai. We HDOA did a survey in August of 2017 at the Hanalei Taro field. On a trial and error basis, because it was our first time with this ant, we used a 3-gram vial with a dab of peanut butter inside the vial. So we left it out for about an hour or so, and we didn't have any luck capturing an Odontomachus. We also did another survey on January of 2020 at Princeville at the Lee Kai Condos for a second reported site with the 3-gram vials. We used cat food this time and still no luck in capturing a Odontomachus ant. With all the storm, floods, and rain we have been having on the north shore and north shore of the island here in the past at Kauai and other work duties and projects, we couldn't have do any Odontomachus ant surveys. Here on Kauai, we find these ants on the leaf litter, nesting in old compost materials and under roots. We also find them about 20 to 30 feet above sea level on the forested canopy. And we find them browsing in pairs or small groups of maybe 4 or 5. Okay, on the right you can see the eggs with actually a pupae with the one ant right here. These we found in the lychee tree, the compost material, which had a pile of ants nesting inside. Yeah, and I have a short video of them on our survey taking peanut butter back to their nests. Yeah. This was at our third site. Yeah. In the LFA survey, we use coffee stirs baited with peanut butter. Is my stuff still on, Greg? Well, in our survey, we see this ant. We see this ant that we notice. We don't want to go in enclosed areas like the small dram vials. We normally use our ant surveys. And this is our areas of location up at the North Shore. it's like 2 miles away because the fire is far apart. Okay, and this is the worker stinger, the male on the bottom left, and the queen. Yeah, you can see the differences in color, but okay. And my acknowledgements. And that's it, Roshan. Thank you.
Thanks, Eric. Yeah, perfect. Slide slow, yes. Yes, perfect.
Good afternoon, everybody. My name is Rosemary Gutierrez, and I work here in Maui as a edible crops extension agent. This work is a preliminary work between the Pacific Basin Agricultural Research Center in Hilo and Maui Extension Office. But as a background, I can tell you that the olive fruit fly is the most important pest of olives worldwide. And the olive fruit fly was first recorded in Hawaii last year in October. First in Hawaii, where our extension agent, Zandal Hamasaki, noticed the damage on the fruits and get the flies and send it to the Department of Ag for its identification. And the Department of Ag confirmed that is olive fruit fly. And posteriorly, we found here in Maui in the same month. So the olive fruit fly is about 1 foot long. The head, the thorax, and the abdomen are light brown color. It has some spots of white and yellowish color in the thorax. It has 2 dark spots at the end of the wings. Also, the larvae is a white, and it has the head is very pointy. Okay. Only 1 egg can be laid by the female on the olives, but sometimes you can see also some big olives with many punctures of sign of a leg was laid. The damage of the olive fruit fly is because of the females lay the eggs on the ripening olive fruit, and the larvae start feeding on it. Besides that, they create an entry hole, and that makes that any fungi or bacteria enter to the fruits and start rotting the fruit. Also, the feeding damage can cause premature fruit drop and reduce the fruit quality for both table olives and olives for oil production. This pest can cause crop losses of 100% on some table cultivars and up to 80% of oil value. We try to monitor the olives fruit fly in Maui and in Big Island in Waimea. In Waimea, Steven Sauder and Nicholas Manowskis did work. They monitor and get the data of the olive fruit fly abundance. And in Maui, I did the work. So what we did is to monitor the flies, the olive fruit flies, we use torula yeast in mag-filled type traps. Okay, we use 3 tablets and we add 300 ml of water per trap. We set up the traps about 4 feet to 6 feet high. This is our traps, the mag-filled type, and some growers did the handmade. They build their own traps to reduce costs. Well, as many growers were very worried about the loss of production, we had to implement a control method, and our PBARC provide us of GF120 to control the flies. Okay, so the GF120 was applied in like plates, put it underside to avoid washing off by rain. So this is how the traps, the monitoring traps and the traps with GF120 was located on the trees. On the right side, some traps with G120 were all washed out by the rain. So something that we need to look for next year, how to avoid the G120 to be washed out. This is how the traps were set up in the field. It was kind of very close because growers don't want to lose production. I want to control as much as possible the olive flies. This is the data of Maui, collected of Maui. We started collecting this data on October, end of October last year. So our first collection, we saw very few ones. The highest one were 5 flies per trap, but many zeros. So, and that was the last week of the harvest of the olives. So we were late for the monitoring, but we keep monitoring the olive fruit flies, and we saw that after the harvesting time ends, and there is no more olive flies in the orchards. We don't know where they go, or they start the dormancy time. We don't know yet that, but there were only zeros until May of this year, where some flies will start showing up on the traps. So, and at that time, the olives were start growing, maybe about ½ inch big, and but there were no damage, no punctures on the fruits, but we got some flies on the traps. And we start applying in GF120 starting, I think, end of June. Yeah. But the population of the fly is still increasing, even with that GF120. On September, where the highest was 20 flies per trap. Okay, but I went this week and there is zeros to no, maybe one per trap in someone's. Yeah, and growers says that damage of olive fruit flies was like 50% less this year comparing to last year. So we don't know how much flies we had last year. Okay, maybe it was higher than this year. And because we applied the G120, that helps to control the population of the flies. So this is the data from Big Island. They start sampling before than Maui, here 1 month before, where in the beginning of September 2019. And the numbers are very high, average of 140 per trap. And as the harvesting time was reducing, the flies also reduce. But there's still some numbers, like up to 20 flies per trap in some sampling times. And in June, start growing again the population. But it's still very low because they use also J120. So comparing these numbers with what they have last year is a big reduction of population with J120. Also, they did an evaluation of numbers of flies in different cultivars of olives plants. So the Museum variety had more flies, female and males, more flies than other ones. And these ones are the table olives. Yeah. And that's why they get the higher abundance of flies. So that's our data for last year and this year of abundance of olive flies. One good news is that Steven and Nicholas found in Waimea Braconide wasp. That was brought, I think, in the 20th centuries to control other fruit flies. And they found emerging from an olive fruit. Okay, this is can be a suggest that if we do a more research on it or work on augmentative release of these wasps, it can help a lot to control this pest and be a component of a comprehensive IPM strategy. In Maui, I didn't have the luck of found this wasp. I am rearing damaged fruits on my office, but nothing came out yet. So thanks to everybody, and if you have any questions, I will be happy to answer. And this is a picture of our field day that we did for the growers in Maui.
How's that? Everybody can see my screen okay and hear me? Good. All right. Well, thank you again for allowing me to come here and update you on this. As you saw from the title, it was originally meant to be a single snail pest new one, but as you all have heard, for insects and other pests coming in, they're coming in so fast and furious that between the time that I was asked to come give this talk and now, we've had another new one come in. So I figured I'd just go ahead and update you on both of them at the same time. And give you a little brief update on what's going on with snails in general. Okay. All right, so just a quick update. This graph here at the bottom left of the screen,
This is sort of our cumulative ongoing updates to non-native land snails in Hawaii. So this includes pretty much all non-marine snails, so aquatic, freshwater, and terrestrial, but dominated heavily by terrestrial species. And the interesting trend that you can see here is that we get a steady increase sometime after the turn of the century, sort of slowly creeping up. But in the last decade or so, we've really jumped way up in this area, both in terms of introduced species and those that are establishing. And I don't know that that's so much an artifact of increased numbers of introductions as it is the increased amount of monitoring that Dr. Yeung and myself have been doing along with colleagues at UH, Rob Cowie, and others over the last 10 years. We've really been doing a lot more monitoring. So of course, we're finding a lot more species. And some of that is just clarifying identities as well, realizing that some of the species that we've sort of encountered and classified as one thing turn out to be 2, 3, sometimes 4 different things. So clarifying the taxonomy is a real big thing there. So those are the 2 things I want to emphasize from that, is that one, we need ongoing monitoring, as you've all mentioned, that we're completely under-resourced to be doing these things. So we have to kind of work together to do these things, but also that we also need to clarify the taxonomy of many of these things to be really clear on what is coming in here and how we're going to control it. And in general, the snails that are coming into Hawaii are pretty much coming from everywhere, but as you would expect, the largest portions of them are coming from places that we do the most trade with. So Asia, Australasia, Europe, and the New World, we're getting lots of introduced things from there. And so currently we're up to about 64 established species. And as you are all aware, I won't dwell too much on this, but they come in through a number of different pathways, but they're primarily agricultural pests, right? Those are the real big concerns that we have for their impacts, but they can also be host to plant and animal pathogens. So they carry viruses, fungal pathogens, as well as nematodes like the rat lungworm and other parasites. And then one of the areas where we're really concerned is with the threat to native species. So not only do they impact native ecosystems and plants, but they're also impacting the endemic species of snails that we have here. And so these are real big concerns of ours. So as I mentioned a few weeks back, we first got contacted by Janice Matsunaga and Darcy at the Hawaii Department of Ag, and they'd found a new snail over in Kaneohe. It was reported to them by a resident there that they had this snail pest in their yard for almost a year now, or probably a little bit more. So we went and checked that out. That's the one on Kaneohe. And then shortly after that, a couple weeks later, we got contacted by a private citizen via Facebook, social media, with another snail from Ford Island. So those are the 2 I'm going to report to you on now. The first one, the one that came from Kaneohe. So that was the site in this residential area up in Haiku area of Kaneohe. If you're familiar with it. And the images here are just some of the shots from the yard where we went and surveyed along with Janice and others from the Department of Ag. And of course, we found a number of specimens, even though the resident had been heavily baiting to try to control them. There's still plenty of them surviving. We found lots of empty shells, but quite a few adult individuals. And from that, we were able to, initial identification was Zachrisia provosora, which is a actionable pest species according to the USDA, particularly here in Hawaii. It's been introduced to Florida as well as throughout the Caribbean. It originally comes from Cuba. The entire genus Zachrisia is Cuban endemics. So there's about 3 species that have been introduced that are all really similar looking from a shell perspective wise. So they're very difficult to tell apart, but this one to us looked like Zachrisia. I did the dissection on this to confirm it, and sure enough, the primary diagnostic feature anatomically is to look at the reproductive structures, particularly the flagellum is much longer in this species than in some of the more closely related species. So the reproductive anatomy matched Zachrisia provosora, as did the genetics. So we also did DNA barcoding to confirm it, and we get 100% match for a specimen from South Florida in the Homestead region. And I just want to put these up here. These are the 2 other species at the top left here, Zachrisia oricoma havanensis, as well as Zachrisia trinitaria. Those 2 morphologically look very similar. Oricoma is the one it's probably most confused with because they're of similar size. The other is a much larger, but all 3 of them cause serious agricultural damage to fruit as well as leafy crops. So they can become a real pest, and they're also known to carry diseases like rat lungworm. But you can see from the three that the shell images are very difficult to distinguish among, particularly someone that's not really well trained in looking at them. The other species that I mentioned, this was brought in by a private citizen from Ford Island. She found this in her yard. She brought it to our attention. She brought us one specimen. We've alerted HDOA. We've had yet the opportunity to go back and survey for these more, but we did get one specimen. Initially, just from the picture, we identified it as Otala lactea. And I just wanted to point out that this isn't the first time Otala lactea has been found in Hawaii. So it actually was found on a ship back in the 1950s. So we have it here in the collection. But more recently, just last year, it was found on a automobile that was imported into Hawaii. Luckily, it was caught in time, and it doesn't appear that anything escaped and established from there. We're probably not so lucky this time. But the images on the right are from HDOA. Where you can see where the snails are hunkering down into the cars, and I'll talk a little bit about that behavior. But I also wanted to point out that and emphasize that this isn't just an agricultural problem. This affects commerce in a much broader way. So this is an article from just last year about the same time of a different snail species, but the same family, that was found in Mercedes cars. So the ones here weren't Mercedes, but in Australia, and they returned the whole shipment back around and sent it back to Germany, and it cost $50 million. So it was $50 million worth of cars sent back just because of snails. So snails aren't just agricultural pests, right? So again, the fastest and most sure way to identify this species is through dissection of the reproductive anatomy. And so that's what I did here, and confirmed that again, it was most likely Otala lactea. It differs again in the length of the flagellum, as well as where the vas deferens attaches to the phallus as well, that location. And there are a few other differences in the mucus glands and some of the other structures. But the anatomy of the single specimen that we have definitely looks like Otala lactea. The other diagnostic feature is that that black lip. So if you look at that shell, the other species that it might be confused with typically don't have that black, dark colored inner lip. Although occasionally there are albino variants of Otala lactea that can be confused with the others. And again, this is an actionable item according to the USDA here in Hawaii. As you can see, there's quite a great degree of color variation, but most of them do have that black lip. The other diagnostic feature with the shell is, if you see in the bottom right, is this lower tooth, what we call a lamellae on the inside of the shell there. So that can help diagnose it as well if it doesn't actually have a real dark black lip. And like all of the snails in this family, or a large portion of them, the helicids, they tend to do this massing behavior. And this is related to avoiding heat on the ground. So all of these helicids, Otala and a couple others, come from the Mediterranean region, so the Iberian Peninsula, Northern Africa, and where they deal with really high levels of heat on the ground. So they have this behavior of during the day massing up on vegetation or any structure that they can climb up on and estivating there, and they're much more active and destructive at night. So this makes them very difficult to detect sometimes, because they'll get into almost any kind of product, as you saw, a car, shipping tiles, containers, anything that's on the ground that they can climb up onto to get out of the heat of the ground and then get shipped around the world. And these have been introduced to Florida, Arizona, California, and even reported in Texas, although we think it's a different species in Texas. We think it might actually be this species in Texas, which you can see looks really, really similar, and actually used to be in the same genus, except it lacks the black lip. And the shell's a little bit different shape, but very easy to confuse with Otala. And then, of course, there's also this one, which is a same family again. This is Theba pisana. This is another one we definitely don't want here. This is the #1 pest in Australia, snail pest. And as you can see from what it does, gathering up on vegetation and things like that, it can be just a pest just because of the sheer numbers. These things get into wheat threshers and combines and things like that, and just really gum up the works and completely destroy food crops so they can't even be exported. So it's a major, major problem. So with that, I just acknowledge a number of people that help us keep track of these things and keep supporting the work that we do here at the museum. And thank you for your time. I'll take any questions if there's time.
Thank you, Ken. So we are already late. So why don't we stay for a little bit longer? Any questions for Ken?
I just, Rashawn, can I clarify? So those were all found on Oahu. For some reason, I thought I was so taken aback by all the pictures of the massive snail populations that I thought I had heard one was found on another island.
No, so they're both Oahu, been introduced to Oahu only so far, but we haven't found them on any other islands.
And is that something that you're fairly confident they're just in those small areas, or should we be looking for them?
Well, I mean, that's a good point. Everyone should be on the lookout for any of these snails. And so just to put a plug in there, anytime you come across snails that you're not sure what it is, or if you haven't seen it before, or even if you have, you're more than welcome to send either myself or Noreen an email with the picture at the Bishop Museum. You can easily get in touch with us and we can say, yeah, that's common, or maybe we should take a look at that. Because as everyone's mentioned, we don't have the resources for all of us to go out there and constantly survey for everything. And so the crowdsourcing of our combined effort is really the important way to detect these things. To get more specifically, these snails, we don't have any evidence that they're introduced elsewhere yet, although we will say that that first one, Zacresia, that was found in Kaneohe, HDOA has worked to track it back to a nursery there where they found breeding populations. So they found live juveniles, and the resident said that it had been around for a year in his yard, so we don't know how spread out it is, and surely it's going to start showing up in other places soon. And I can't emphasize how bad these two are. So both these species are horrible, horrible agricultural pests. I mean, they'll eat almost anything, and they'll do it in an incredibly voracious way. They can amass numbers of millions very quickly. And can be a real big problem. So they're going to be on the order of, like, giant African snails or Veronicella cubensis, or the leatherly slugs. They're going to be that kind of pest that we're going to have to really work to control. The one on Ford Island, we may be lucky right now because it's Ford Island. It may not have escaped Ford Island. It may be restricted to the residential area, and we might be able to eradicate it. I know Darcy is working with David Robinson at the USDA to develop an action plan for Zacresia and Kaneohe and with the nursery to try to figure out how far it has spread and sort of develop an action plan. But one of the things we think would be really good, and the reason we wanted to do this, was to get the word out there for people to be on a lookout for this so that we can sort of stamp it out in pockets if we find any of these and any other species that we need to take action on.
Do you have any printed materials or something with the ID on those? Just we could share that around. We're doing a class coming up in the new year, teaching teachers how to work with their students to collect slugs and snails, and obviously we always send in anything. I just sent something to Nori not that long ago, actually saying, hey, what is this? But something that we could share with, like, those folks to say, hey, there's these potential new things that you should be on the lookout for, just so they understand that this is an ongoing issue.
Yeah, so as a matter of fact, we're working with a student at Iolani that's developing a phone app, for one, to help identify non-native snails, which is going to be great once that gets out. And we're also going to mirror that on a website that will help. But we do have some materials, and so if you'll get in contact with Nori and I, we can discuss the details of what we can get you and specifically targeting what you need. Because we have a lot of curriculum that Nori and I have developed over the years to help with those kinds of things, to help with training and sort of snail taxonomy and sort of the urban ecology of invasive species like that. So we're happy to help provide that information.
That would be great. And then can I get your email in the chat? I missed it on the presentation.
Sure.
Sorry. I was really wowed by those snail pictures.
Sure, no worries. Well, I wanted to go quickly. I knew we were running behind, so I wanted to rush through it sort of quickly without boring anyone too much. Here, you can reach me here.
So Ken, there is a last question. You can
Sure.
also address that question quickly. Erin Bishop has a question in the chat box.
Oh, I see it. I see.
Okay.
Hey, Aaron. So that's kind of why I was sort of hesitating a little bit, because Darcy and them are working up their plan to get the information out there. We really are trying to push Kevin and Darcy and them to really move quickly to broadcast this information and get the details out so that we can make sure we stamp this out, particularly the Zacrisia, before it gets too spread out. I'm hoping there will be a pest alert. I'm hoping there will be sort of a broader effort, but I think I'd have to refer that directly back to HDOA as to how they want to handle it. I mean, again, we're Bishop Museum taxonomists and researchers, and we do the research and the identification, and we understand the importance of it, and we want to see it done, but we typically have to defer to those that are in charge of doing those things. We don't want to step on anyone's toes, and we were told to be very careful about what information we disclose.